Hello. Let's see who else joins first. Right. If you are here for the year 11 stuff that I do every day, I will be doing that specifically a bit later. If you have any questions, then put them in. But the first part of what I'll do is there'll be people turning up to ask some homework questions, which I will answer first. But I will start that in. I usually start at 7:15. But if you have any questions, um, put them in so that I can be ready to do them. And I'll be back in a few minutes. Hello. Right. I'm going to start by answering the questions that have been asked from the Year 7 booklet, which is this one. Um, if you have any other questions, whether whatever year you're in, just ask them now. I know some Year 11s might be joining now as well. Just ask. If there's anything specific, I'll get round to it eventually. But otherwise, initially, I will just get through for the remaining sessions that we have until the sort of the next month and a half. It'll be quite quick just to get through these questions that I need to focus on the Year 11s who will probably be joining for exams. Right, but all of this is still relevant to everybody, so watch along. Cells are the building blocks of life. Draw one line from each type of organism, the diagram of one of its cells. Um, an animal cell. Ooh, okay, well, let's start with plant. I think that might be slightly easier. That looks like a plant cell to me because I can see the cell walls there. Um, and then, and the only animal cell here is probably just this one because it has the features of an animal cell, a nucleus and a cell wall, cell membrane. And a bacteria cell will often look a bit like that. It doesn't have a nucleus, it has the bits around the outside, so that will be that one. The test should be marked or about to be marked, so hopefully this weekend. It takes a bit longer usually to mark the tests because just of the nature of it. Um, but cells contain structures. These structures have different functions. Draw one line to each. This goes back to the topic of cells. Contains genetic information is your nucleus. Controls what enters and leaves the cell is a cell membrane. Where photosynthesis happens is a chloroplast. Um, 136. I'm still on one, aren't I? God, it's so big. C. Choose a chemical produced in cells. A chemical excreted by animals. Out of these, the chemical that's excreted is probably urea. Urea is the stuff in your urine, so it'll be urea. Um, Stored in animal cells is this stuff. You probably wouldn't know that, to be fair, so it's good to do that. When we store sugar, when you eat sugar and it's stored in your body, it is stored as something called glycogen. Um, Plant cells store starch. Similar thing, but different. A chemical that strengthens plant cell walls is called cellulose. And that's that. That is an animal cell. Give a function of muscle cells. The function of any muscle is to contract um, to help you move. And I think contract would be just enough of an answer to that one. Yes. How are muscle cells adapted? There is a clue in this question, which is labeled, which is all of the mitochondria are labeled. Hopefully, you know what mitochondria are responsible for. Mitochondria help to release energy from respiration. So, the way muscle cells are adapted for their function is that they've got lots of mitochondria, as we can see here. Oops. Which help it to release lots of energy. That's it. I'll get to that booklet three. Any other questions from any of the booklets, please tell me quickly so that I can get on to other stuff should anybody need help. Um, right. One muscle cell was this wide. When viewed using a microscope, it was 2 millimeters wide. Calculate the magnification. So there's in this sort of formula, there is something called an image, which is what you see down the microscope, which is 2 millimeters wide. And there is the width of the real thing, which is much smaller, 0.05. And if you divide those two, it gives you what's called the magnification, which in this case is 40. Yeah, booklet three, I will get to. I've got quite a few to get through first on one, so I'll get to that first. Um, Question three. Complete the equation for photosynthesis. Carbon dioxide plus water gives glucose plus oxygen. That is the thing there. The energy for photosynthesis comes from light, and energy is absorbed by a green pigment called chlorophyll, not chloroplasts, chlorophyll. Um, and if you make it colder, photosynthesis happens less, basically. So it will decrease if you make it cooler. Um, and then three ways in which plants use glucose if you are here in year 11. And then this is a very typical question that you get about how plants use glucose. And you should have a few answers in your mind that you're about to see. For example, the glucose is used for respiration. respiration requires glucose. Um, it is also used, or they store it as starch, or used to make starch for storage. And it is also used to make that cellulose, which is what the cell walls are made of. There are other answers to the question, but I think these are the easiest ones to remember. Um, so yeah, let's stick with that. 3, 6, 7, 8. Six, antibiotics. Which substance is used as an antibiotic? Out of these three, penicillin is the antibiotic. Um, symptom of gonorrhea. So this is a STD. So you want to say something like a discharge, which is like an unwanted fluid being released. You could mention something like just um, pain on urinating as well as an answer to that question. Okay. Um, right. They investigated which antibiotics were most effective. I've seen this before, so I'll just continue. Two control variables. A control variable is anything you keep the same every time you do the experiment. So often there's a bit of clue in the method. They say they placed them in four different antibiotic solutions of the same concentration. That's something they've kept the same for all of them. They've also put used four discs of the same size. That's something else they've controlled. They've also kept everything at the same temperature for the same amount of time. That's also a control variable. It's things we keep the same. Booklet 2, 3, see, I'll come to in just a moment. I think a couple of these will be the same. Um, but, um, and then what they did was they did it with two types of bacteria, gonorrhea and chlamydia. And you can see, based on the circles, which bacteria were killed. Which antibiotic did not kill either type of bacterium? Well, B, because there's no circle around it. Um, B, because there's no circle around it. So B. Which antibiotic would be the most effective to treat a person with the gonorrhea infection? It's the one with the biggest circle around it. So gonorrhea is this one here. That looks like C with the biggest circle around it. Um, I think booklet 1 and booklet 2 are the same, actually. I think they are. So we'll see in a moment, depending on the questions. Which antibiotic would be most effective with both gonorrhea and chlamydia? Probably A, because A's killed quite a lot of bacteria, hasn't it? Um, antibiotic cannot be used to treat HIV. HIV, the V tells you it's a virus. And antibiotics only kill bacteria, not viruses. Fungi can cause an infection of the fingernails and toenails. It can spread. Some people go to nail salons to have their nails shaped and painted. One way in which nail salons can reduce the risk of infection is probably by wearing gloves, I'd have thought. I feel like a sensible answer. Um, yeah, new gloves for each customer. You don't just say wearing gloves because if you wear the same ones, then that doesn't work. Or you could disinfect your tools between customers, disinfect your hands between customers, use new gloves for each customer. That would be a sensible thing to do. Wearing gloves doesn't make sense if you think about it, just wearing gloves. Because if you wear gloves and you treat somebody, and then somebody else comes along and you've still got that infection on your hands, on your gloves, and then you treat them. So the point is you need to change your gloves, like subway workers. Um, so that's one reason why fungal infection of toenails is more common than fungal infection of fingernails. Um, God, I think it's because your feet are in warmer conditions, usually like in your shoes. They're also generally washed less frequently than your hands. I think that's probably the best answer. Okay. Um, and then what was it? Six, seven, and eight. So I need to get through this quickly. Um, give two ways the body protects itself from these. I'm going to say these now rather than write them down. But the ways in which your body protects itself from these things on your skin, you could say something like, we've got the skin itself is a physical barrier. It's stopping them from getting in. You could mention that anything that does get in gets killed by your white blood cells. So two ways in which you protect from these microbes. These might, if they get into your body, white blood cells kill them. So these like little things on your skin, basically. Um, and eight. Bear in mind, by the way, that everything that I'm doing gets recorded, so you can re-watch it. 6C was same concentration, same size disc. It's basically where look for the word same in the method, and you'll get the answers. Bacteria and viruses make us feel ill because they produce toxins is the answer, even though viruses actually do something else. Um, Antibiotics are medicines that kill bacteria. And then new strains have developed which are resistant to antibiotics. New strains are produced when pathogens mutate. Um, yeah, I think that's what it is. What will scientists have to develop to kill these new resistant strains? They'll have to develop new or stronger antibiotics. So basically, our antibiotics are always getting a bit stronger because each one, eventually the bacteria evolve and become resistant to it, which means that it doesn't kill them anymore. And then we try stronger ones to kill the new ones, and it kind of keeps going in a cycle. Um, right, that's 678 on the answer to other questions. I'll get to booklet three. There's one question from booklet two. I'll just get that done. I think we've already answered most of these. Yes, the ways in which plants use glucose, I'll just repeat. It's the same question from earlier. It is here. So plants use glucose for respiration. They use it to make starch, to store it for when they can't make the glucose, like at nighttime. And they also use it to make this stuff called cellulose, which is for their cell walls. That is the answer to that one, which will move me straight on to um, booklet three. Okay, 2I and 2 part 2. Okay. The table shows changes in resistance to the antibiotic penicillin in one species of bacterium between 1991 and 1996. So as you can see, as time's gone on, more and more or bigger percentage of this penicillin has become, or this bacteria has become resistant to penicillin. Um, booklet 2, 1E and F. Right. If you have any other questions on booklet two, then you need to ask them now, not separately, if that's all right, because I need to move on. But I have actually answered both of these questions in the video earlier because booklet one and two were the same. So I've actually answered those questions. So if you go back and watch this, then you'll have the answers to that. Um, so booklet three, 2I. How does penicillin help to treat an infection? Well, it kills bacteria. That's the first thing. It's an antibiotic. So all antibiotics kill bacteria. Um, and part two, use the data to say why the doctor should not prescribe penicillin. Well, because it seems as though these bacteria are getting more and more resistant to penicillin. That means that the more you prescribe it, um, the more they get resistant to it. And so we probably maybe should not prescribe penicillin for this because quite a lot of these cases suggest that this bacteria is resistant to it. So the simple answer is the bacteria might be resistant to penicillin or it won't be killed. So it might be resistant, so it won't be killed. That's one mark. And secondly, it may actually, well, I mean, the best answer to the question is that it seems as though I'm trying to think what the best answer is. The best answer is probably that if we keep on prescribing this, then the penicillin will get less and less effective because over time these things get more and more resistant. So it might already be resistant, but if we keep prescribing it, then the penicillin will get even less effective. So it's already 22%. It might be like 30%, then 50%. You'll keep on increasing how resistant this bacteria is. Okay. 3A, I should not need to do. I hope nobody asked what 3A. No, I haven't. Good. Um, I'm not going to do a question about vaccinations. 3B, um, and 3C. So 3B and C. Oh, let me just check something. I'll have to come back to that later. Um, in the 1990s, many people thought the MMR vaccine caused autism in some children. This is why the Japanese government stopped using it. This gives information about the percentage of Japanese children who developed autism. Okay, so these bars show the percentage of children vaccinated, and then the line shows the number of children who developed autism. Okay, there's no link there at all. Um, the data supports the view that there is no link between that. Explain why. Okay, well, um, the easiest point here is that in 1996, roughly, when there were no vaccinations, or between 1995 and 1996, when there were no new vaccinations, the number of children who developed autism increased significantly. There was a big increase in a year when there was no um, vaccine. So you can mention the fact that um, the percentage of children vaccinated was zero in 1995. So put this in your own words, what, zero in 1995. And yet the number of children developing autism rose a lot. You're trying to prove that there's no link between the vaccine and autism, which is what this data suggests, right? If there's no vaccines, yet there's a huge increase in the number of people with autism. It suggests there's nothing to do with it. Okay. Um, that would be a couple of the marks. And I would just say that the highest, I would also mention this, the highest number of children with autism happened when there were no vaccinations. Okay, which suggests that there might be some other reason that these children are developing autism. It doesn't seem to have anything to do with the vaccine. So say that as well. So it suggests that there's some other reason for these children developing autism other than the vaccine. That is 3B. Um, okay. Right. 4E, when you're doing this experiment, how could you make the results more valid? Don't mention the temperature. Difficult question until you do all the required practicals properly. But I shall tell you is that pretty much any question that says, how do you make this experiment better? Usually an answer is that you can repeat it and calculate a mean. So rather than just do it once at this distance and count the bubbles once, why not do it 3 or 4 times and calculate a mean? And second point I would say for this investigation is that if you've done this, and some of you will have done this, where you basically count the number of bubbles you see like for a minute. Now, counting bubbles is not a great way to measure this because sometimes you can't see a bubble, it's very, very small. A better way to do it would be actually to connect something called a gas syringe to this thing, which will give you a more exact measurement for the gas produced. These bubbles are just bubbles of gas. You're just counting bubbles. It's not very good because one bubble could be bigger than another and stuff. Um, but this way, you're measuring the actual volume of gas produced. That is a better way of measuring the rate of photosynthesis. So the best answer here, apart from that one, would be to measure the volume of gas produced. Is there anything else I could say would be easier? Use the same lamp is apparently an answer. Okay. Um, right. I'm not going to answer the last question that has been asked, 5, D1, D2, and D3, purely because you didn't ask it early enough. Um, which suggests to me that you're looking at it right now. 2, 4E. Oh no, you did. Yeah, not on Austrian. 4E and G. We still on 4. Where's 4? What did I just do? Okay, E and G. Did E, right, G. Um, Give a conclusion that can be made from the table above. The conclusion is really easy. It shows that as the distance of the pondweed increases, the number of bubbles decreases. That's just saying what you see for that question. Um, 5B, two things that can happen in the plant are the glucose produced in photosynthesis. I've already answered this question. The glucose can be used for respiration. It can be used for storage as starch. It can be used to make the cell walls, which are made of cellulose. Um, Plants need minerals. Through which part do mineral salts get into the plant? Explain why water is important. Ugh, it's quite a hard one. That will be to do with osmosis. Yeah, so why is water important to get mineral salts into the plant? Most of the minerals are dissolved in the water. They're not just on their own like little pieces of mineral. Minerals are often dissolved in the water already. Um, and the water is what transports the salt throughout the plant. Um, Probably that. Um, and then, yeah, I won't do that. Um, deep. I mean, that should be an easy question. Um, I can tell you that nitrates are used for making proteins. That was a slightly difficult question. That's what nitrates are used for. And is there anything else? 3B I've done. Yeah. And then I'll have a quick look at this one. Um, 4B on booklet 2. Tomato growers alter the condition of the greenhouse to make tomatoes grow faster. Which change in the condition will make tomato? So what you're trying to do for a plant to grow as fast as possible, you want to increase three things: the temperature, or get the perfect temperature, increase the amount of light, increase the amount of carbon dioxide. There are three factors that affect the rate of photosynthesis. Um, so temperature is an answer. And turning the lights on at night, that increases the light. That should be the two answers for that question. Right. Given that there are no other questions, um, I will stick around here for a little, well, I'll be back in a little bit because there might be some people from year 10 asking some of the questions on the year 10 homework. If you are here from year 11, then again, just if you have any questions, ask them. Otherwise, I'll be going through some homework questions. Presumably, if anybody comes on and asks, which are all quite complicated questions on circulation and digestion, which, if you're in year 11, you have an exam on in 6 days. Um, yeah, fine. Nothing else? Nothing else immediately? Okay, fine. I'll be back in a bit. Hello. Right. I'm go right. I'm going to answer the questions on here. For those of you in year 11 that might be here, um, these are all topics that you are likely to have depending on your exam board in your exam on t- 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 next week. Next week. If you are here, just so you know, if you're new here, then if you're in Key Stage 3, then you want to join between 7 and 8 and ask any questions on the homework that we set. As I was going through earlier, at this point, I will mainly focusing on the Year 10 and Year 11 students. So, all of question 1, let's have a look at that, which is this one. Um, a student pedaled an exercise cycle at constant speed for 5 minutes. The student's heart rate was recorded at 1 minute intervals during the exercise and also during recovery. So as we can see, as you're exercising, your heart rate's going up, and then you stop, so it goes down, and it comes back to normal. Describe as much detail as you can the changes in heart rate between 0 and 14 minutes. Okay, so make sure you identify which bit you're referring to. Um, um, yeah, if you haven't got your homework yet, then hopefully it will be due to that reason that it might have just been delayed, but it's all right, you'll get it, and then you can deal with it then. Um, um, Describe the changes in heart rate. So I would mention first that it initially, when you started exercising, it rose rapidly. Also, the person who mentioned that all of the questions on year 10, if you just let me know who you are, because it's quite important that we know, um, rose rapidly during exercise. Then I would normally say in this question that it reached a maximum, like mention some numbers, like it reached a maximum of whatever that is, 126. So reached a maximum of 126. After which, what happened? It fell quickly during the recovery phase. It fell quickly um, for about, what's that, 5 to 7 for a couple of minutes. See, a steep line here shows it falling quickly, but then it continued to fall, but it fell more slowly then, didn't it, for a few minutes? So this bit here, because the line's not as steep, it's fallen more slowly. Um, so then it fell more slowly for 4 minutes or between 7 and 11 minutes. And then I would say something like it returned to its original heart rate after about 11 minutes. Something like that should cover those up. I know that's you, sorry, the person who was wanted me to do all of this booklet, which is optimistic. Um, so how do arteries supplying the leg muscles alter the rate of blood flow through them during exercise? Well, during exercise, would you agree that your leg muscles are working harder? It's quite hard to get this mark actually, which means you want more blood to get to your leg muscles. So the arteries that are supplying this, they widen to let more blood flow to those leg muscles. So something about widening would be what you want to say here. Um, okay. Explain how an increase in heart rate helps the student during exercise. Now, this is a very common question, and the answer is basically always the same for this kind of question. How would an increase in heart rate help you during exercise? Well, during exercise, your muscles work harder or need more energy or use more energy. If they use more energy, they need to respire more or more quickly. Because remember, respiration is what releases energy. If they need to respire faster, they need to be supplied with more oxygen. So a faster heart rate supplies more oxygen to those muscles because it's beating harder and faster. Um, and also, there's two things we need for respiration, right? There is um, glucose and oxygen. So it supplies more oxygen and more glucose to those muscles. So the heartbeat beating faster pumps your blood to where it needs to go more quickly. so that you can get, crucially, oxygen and glucose to those places, so that those places can respire more quickly to release more energy. That is the sort of common sort of question here. And you can get asked it the opposite way. Like, for example, if I said, how would smoking affect you during exercise? You could mention that your lungs get damaged, so less oxygen is supplied to your um, muscles, which means they respire less quickly, they release less energy. You can sort of turn it around and change the question a little bit. Um, Let me just answer the question that's specifically being asked. 4B. That's the same question. So the answer's the same as well. Which is that the heart rate supplies more, that you get increased blood flowing around your body, so more glucose and oxygen gets transferred around your body. You get faster respiration, more energy released. That's it. It's the same question. Okay. And all of 27. Was 27 the thing at the end? Yes. Um, right. Figure 3 shows the activity of the enzymes pepsin and trypsin. Activity when it comes to enzyme is just how quickly they're working. Okay. So describe the trend for trypsin, which is this one. So you should say that for trypsin, um, it's only the enzyme activity initially increases from 5.8 to 8, right? So it's increasing when the pH is between 5.8 and 8. So activity increases. That looks like 5.8, doesn't it? Then I would mention the fact that this is the optimum pH. So the optimum pH is at pH 8. Um, and then I would say that after pH 8, the activity decreases, or it decreases between 8 and 9.8. Quite obvious when you see the answer written down. You're just saying what you see. And for those of you that have important exams coming up, a describe question just tells you to say what you see. It's not asking you why this is happening. If it said explain, then you need to explain why it is happening. Um, so yeah, I will do more questions from this booklet. It's fine. I'll just focus on this because there probably aren't any other questions, at least at the moment. State the optimum pH for the enzyme pepsin. Pepsin is there. The optimum pH is where the graph is at the highest point. So it's at pH 2. And pepsin only works effectively in the stomach. Describe the conditions in the stomach that allow it to work effectively. You should know that the stomach is acidic. um, because it produces hydrochloric acid. And they are the 2 points. And that is why the conditions in the stomach are good for this enzyme, which works best at pH 2. In other words, in acidic conditions. Okay, that is all of those questions. Okay, I'll have a look at some of these questions and decide whether they're worth doing. The name of the enzyme that digests starch. We should know that there are three enzymes, amylase, lipase, and protease, and you should know what each of them digests, regardless of what year you're in. Um, where are most food molecules absorbed? Um, I'm on question 2 at the moment. Um, the most food molecules are absorbed in your small intestine. So your small intestine goes like that. There's millions of these little structures called villi in your small intestine, and they absorb the food and put it into the blood. Small intestine. Um, these show two villi, so those little structures that I just said. Okay. Um, and they say, what's the advantage of using an electron microscope? An electron microscope has a higher magnification than a light microscope. It zooms in more. It also has a higher resolution, but it's only a 1 mark question. What type of blood vessel is labeled X? So wherever you have things moving in to from one place to another into the blood will be traveling through what's called a capillary. A capillary is a blood vessel or a pipe, but it's got very thin walls and it's used for when things have to go in and out of the blood. Um, okay. And then this question, again, it's really a maths question. The question tells you the magnification is 20. It says the real size is 0.8, and it says calculate the image size. So if you call that x, and I said do this as a maths question, you would hopefully do 20 times 0.8 and get 16. I think that's it. Yeah. I'm not going to do that question, but I will do that. Explain how villi are adapted for efficient absorption. You should say that villi have got thin walls, which reduce the distance for diffusion. Diffusion is the movement of particles from a high to a low concentration. And if they're going through a thick wall, it takes a lot longer. But if they're going through thin walls, it's quite nice and fast. You can also say that they have a large surface area because there's only two of these in this picture, but there's millions of these little things. Um, and by having so many of them, they have a large surface area, and a large surface area maximizes diffusion. And I'm only going to put those 2 points because I think that's all you need. I'm not going to answer that, or answer that, or answer that. Alveoli are adapted because they have thin walls and large surface areas, the same as villi in terms of the answer. A gas which is excreted means removed, which gas is removed from your body. Hopefully you know that. The name of the process of respiration when oxygen is not available is called anaerobic respiration. And in human beings, I think the main thing you need really is so describe the process of respiration in human beings when oxygen is not available. So it's asking you to describe anaerobic respiration in humans. The equation, which is probably gets you a couple of these marks, is glucose arrow lactic acid. Lactic acid is what's produced from anaerobic respiration in humans. And I think that's pretty much it, actually. Describe the process of respiration. Um, and I was mentioned something like it releases energy, but less energy is released in anaerobic respiration compared to aerobic. I'm not checking the mark scheme. I'll just tell you what you need to know here. An artery is a blood vessel that carries blood away from the heart, which means you should be able to figure out, um, based on the arrows, what that is. Oxygenated blood is generally in on this side of the heart. Okay, so the left side of the heart, but the right as you look at it, that's the left side of the heart. And then I've already answered this question earlier in the um, well, I'll answer this question because it was the same questions earlier. And then I'm not going to label the digestive system. How protein is digested. I mean, yeah, this is all stuff that this is irrelevant that this is a year 10 booklet because all of these would be exactly the same questions, pretty much. Um, Protein is digested by an enzyme called protease. That's the name of the enzyme. It is digested into something called amino acids. Um, and maybe where it's produced in the stomach and the small intestine. Um, and bile is an important question to mention for everybody, really. Bile is a substance which emulsifies fat. Emulsifying, it means that it increases the surface area of fat molecules, which means that they get broken down more quickly. And also, bile is an alkali, which is helpful because when you get food into your stomach, it has acid all over it. So what we want to do is neutralize it, and the alkali helps to neutralize the stomach acidity. Okay, I think that's probably enough for that. Um, the shape of a red blood cell, you could mention that the shape of it, it has a biconcave disc shape, which gives it a very large surface area, and its job is to carry as much oxygen as possible. That allows it to absorb more oxygen. Okay. Hemoglobin binds with oxygen, what it does. And if you want to just a subsequent mark if you need it, it forms something called oxyhemoglobin. And I think that pretty much covers it. Right, I've got a question from the year 11 one, unusually. So let me do that. If you have any other questions, then let me know, but I think that pretty much has covered everybody. Um, I need to just focus on the year 11 stuff. I think this was what we gave. I haven't actually looked at this at all. That's three gone. Okay. Four. This one, is this what you're talking about? Yeah, 4E. Okay. Describe the relationship between age in years and time for the muscle to contract. Isn't it that as you get older, the time for the muscle to contract increases? That would be one mark. But the second mark will be that at higher ages, the difference gets bigger. You see how there's a difference here of about 1 second, and here it's like 3 seconds, then it's about 3 seconds, then it's like 5 seconds, then it's like 6 seconds. So the difference is going up as well. So when there's a 2 mark question on this kind of stuff, just think about um, yeah, just think about the fact that there's 2 marks here, so you might need to explain a bit more. Proportionally, I don't think you can. I will just check what the mark scheme says because it's not proportional suggests that it's going up by the same amount every time. I think it's literally the opposite of proportionally. Um, at an increasing rate. It says, and the mark scheme says, do not accept directly proportional. So you need to mention at an increasing rate, or say that at higher ages it goes up by more, something like that. Because proportional would mean, just so you understand, is that at age 30 to age 40, the difference is the same as it is between 40 and 50. That's what proportional means. So this difference is the same, but it's not the same. That's the point. It is um, an increasing rate, which means the graph of this, if you plotted it, would actually look like that. That's not proportional. Um, that is, yeah, that's not proportional. It is a curve. It is a faster increase at higher ages than it is at lower ages. Um, okay, what else was asked here? 5E fossils. Ugh. Okay. Um, um, well, you need to know the three-domain system of classification, which I appreciate for your example. You possibly don't know, need to know yet. Um, but I believe it is the eukaryotic, prokaryotic, and then there's a third one called archaea, which is like extreme things. And so fungi are not in that one, so they're going to be either prokaryotic or eukaryotic. They're not prokaryotic, so I think eukaryota is the answer. A bit far away, this isn't it? I'll do 5C thingy in a moment, if I can find it. 5G. Um, give three types of evidence that are used now to classify living things. Do not refer to appearance in your answer. Um, Jeez. I'll tell you what the answer is. It's not an easy one, this, but studies of their cell structures with light microscopes. For some reason you meant to mention the light microscopes here. Um, DNA analysis seems like an obvious answer. Like that helps now. We can check things like DNA and stuff to classify things properly. Um, and another simple answer, a simple-ish answer would be study of evolution. So we know that something's evolved from something else, which allows us to classify things appropriately. Um, 9 part 3. Oh, I've got some pages missing. Here, isn't it? Earth is happening here. Um, so in a different experiment, a sucrose concentration of 0 increases the mass of a carrot chip by 30%. So 0, there would be if you imagine drawing the graph again, at 0, it would be at 30%. And then it says that for the carrot shows a 10% decrease in mass compared with its original mass for every zero point 0.2 increase. So at zero point 0.2, it will be 10% less than this, which will be 20. Oh God, how would you do that? Oh, sorry, 10% less. Sorry, yeah, it's already in percentages. Um, so a 10% decrease in mass for every zero point 0.2. So that would be then 20%. Then at 0.4, it would be at 10%. And then at zero point 0.6, it would be at 0%. And I think the question is, um, the carrot chip should, if you drew that graph now, calculate the x-axis intercept, 0 0.6 must be the answer. Yes, it is. So essentially, you're following the path of the graph based on what it says there to get there. Okay, let me go back to year 10 and 5C part 2, see if I've already done. I think I've probably already done it. Oh, of course not. Okay, right. Salivary amylase. They collect some amylase, put a different pH solution and some food in each of 6 test tubes, added some amylase, recorded the amylase activity after 10 minutes. Okay, that's starch. Suggest what happens to the breakdown of the substance when food reaches the stomach. Well, according to this, in acidic conditions, the enzyme has zero activity. In other words, it does not work in acidic conditions, according to this graph, which we should know already, but according to this data, we know as well. So, what happens to the breakdown of this substance? In other words, breakdown of starch? Well, it stops. Starch stops being broken down in the stomach because the enzyme activity is zero. Let me just check precisely what we want. Um, and it's because the stomach is acidic. And we know from this graph that amylase does not work in acidic conditions. What about the 10%? I'll go back to that now. Um, so this 10%, I assume you mean? Well, that's what I used. I used the fact that they had already told us that at 0, it was 30%. And then it said for every increase of zero point 0.2, this dropped by 10%. So that's why I put from 30 down to 20 at zero point 0.2. And then 20 down to 10 at zero point 0.4. If you drop that another 10%, you're at 0 and you're at 0.6. And that's the x-intercept effectively. That is what that means. Does that make sense? Mhm. Just as a reminder again for the few of you that might be here in year 11 at this point, reminder that these the idea of this is for you to ask questions if you want me to cover something or explain something or do a question and send it. Then this, I presume you're coming across lots and lots of questions all the time at the moment. Um, you should be finding things that you don't know how to do unless you're going to get a grade 9, which is great. Um, but otherwise, there should be things that you feel like you could get ask for help on, and that is the purpose of this. Um, okay, fine. I'll sort that interpolation thing out. As in, I did the example, but I appreciate that was very quickly done the other day. We'll do it another day in between now and then. Just to make sure you get it. It is very easy. It's just a weird process. Um, fine. Right. I don't think there's anything else for today. So again, for year 11 students, on this same link that you are on, I will be here tomorrow at 7 p.m. Not on Friday because I have to be teaching at 7 to 9, but then Saturday, Sunday, Monday, Tuesday, Wednesday, like every day. And then next Monday night, um, before biology paper one, I will just do a longer session going through the whole AQA content, and that is the intention. Um, Question three on what paper? I don't know who this is asking these questions. Have you covered Q3? Um, no, because I didn't feel like I needed to. This one, this question. Again, I still don't know who this is, so if you could just let me know who you are asking these questions. Okay. Right. Anyway, on the basis that there's no more further questions, I think I'll leave it there for today. So again, once again, if you're in year 11, every day available, 7 p.m. ish for a little bit, and then next Monday night I'll do a longer one, and then next Wednesday night I'll do loads of non-calculator math stuff, assuming you have that, or whatever one you've got, and then same again before every exam we will do some longer stuff to cover the content, mainly for AQA for science because it has to have to pick one. I seem to be the most popular one. Right, I think that's it. So I'll leave it there for today.